Kristen Atchison here, and we're talking about reading research articles more than you ever wanted to know. Now, reading research articles by themselves can be rather boring, so I'm sure a video lecture at that, on reading research articles will be mind-numbing. So I apologize ahead of time, but it is what it is. Again, there's some nuances to research articles that are different than how you read textbooks and how you read fiction and these sorts of things that are kind of important. So I'm going to go through this. Feel free to kind of go to the sections that are really relevant to you if you need to. If you're really comfortable with reading research articles, maybe this isn't the video for you. But I'll do my best to keep the energy up so that nobody falls asleep. Okay, so the structure of a research report has a title page. And when you write a research paper, your paper will also have a title page. The APA submission guidelines, which is what APA format is all about how to submit a manuscript. And you're going to adhere pretty closely to that. So a lot of the things that we're going to talk about that they do are going to be similar to the things that you'll do. There will be differences because you're writing different kinds of papers than they are. But some of the APA stuff will be very, very similar. So there's a title page, an abstract. You guys are not responsible for an abstract. Introduction, methods, results, discussion, references, appendices, author's note, footnotes, possibly tables, possibly figures. This part right here, the introduction, methods, and results discussion, is really the body of the paper. That's really where all your important information is. And they have these subheadings to really tell you kind of where to go. So when you give me a research article and I'm looking at to see what kind of research design it is, first I look at the abstract. If that doesn't tell me, then I go to the methods. I know that's where that information is going to be. I don't have to read the whole research article to decide what kind of methodology it is, because I can start at the abstract, and if that doesn't work, go to the methodology and look just there. So that's kind of the advantage of kind of giving you these subheadings, is you can kind of figure out kind of what is going on. The introduction really sets up the problem. It tells what the research question is, what the researcher's hypothesis is, and that hypothesis is, again, the prediction of what the answer is going to be to that research question. Okay, it's what their best guess is, what the answer to the research question is. So remember that hypothesis and that research question are different things. But all that information is typically in the introduction. And also, these research articles are kind of a conversation between scientists. And so this is the introduction will also reference kind of where we were in the conversation. This is what we know so far, but we don't know this, our research question. So I think this is going to happen, hypothesis. So they're going to have this kind of information in there. The methods, we'll discuss the methods, results, discussion, etc. And we'll talk about each more of these in detail. The appendices, author's note, footnote, table, and figures, these really are dependent on the individual author writing and what need that they're having. None of your papers will have any of these things, okay? So the structure of the paper, it's kind of like an hourglass. It starts out very, very broad at the introduction. It gets very, very specific towards the middle with the methods and the results. And then it ends up, again, broad at the discussion and conclusions. So again, we start with that broad problem, our research question, our research ideas. It tells us where we are in that conversation already. And it tells us what we think is going to happen, what we think that prediction is going to be. The method gives us the details about how the study was done. This is specific enough to allow replication. So again, there's going to be details in this that aren't important for a summary, because a summary is not about replication. If I'm interested in replicating that study, I'll go to the original source. A summary is not going to have that level of detail. A summary is going to stay a little bit more broad. Results. This tells us what they found. Given their methods. Basically, this is going to tell us, is there a difference, okay, in the groups, okay? Because again, we're primarily looking at experimental designs here. So was there a difference between the levels of our independent variable? That's what the results are going to tell us. Yes, there was a difference, or no, there wasn't a difference. And again, this is very specific because they're talking about individual numbers. The discussion is going to start to get broader again, and instead of talking about these individual numbers and these sorts of things, they're going to start to say, what does that difference mean? Okay, so the results in discussion are different because the results will say there is a difference or there's not a difference, and the discussion will say what that difference or what that lack of difference means. So it starts to get broader again. And that conclusion, that last piece, is going to kind of have those take-home messages. Sometimes this is included in the discussion, but it's going to be kind of the implications of the results. So results are going to be very specific. There was a difference, there was not a difference on these specific measures. The results are going to say what it means that there's a difference, that the independent variable caused change on the dependent variable. That's going to be in the discussion. And then the implication and the conclusion is going to be, okay, so we saw a difference between treatment and control group. We saw a difference in the two in different groups of the dependent variable in results. The independent variable caused change on the dependent variable, discussion, and conclusion is going to be what are the implications? So the independent variable changes the dependent variable. That means X, Y, or Z in this broader scope. These kind of overarching take-home messages. So the introduction, again, what problems being studied, what does it add? Again, this is where it's going to have the kind of where that basis for the hypothesis came from. Nobody comes up with a hypothesis just kind of out of the blue and then studies it. We go and look and see what we've already done. What do we already know about this topic that we're interested in? And we go and based on that information, based on this previous literature, we make our hypotheses. So again, they're going to give us that introduction. In that introduction, they're going to give us a kind of a recap of the literature up to date that's relevant to the question that they're asking. There's primary objectives in this. They're going to introduce what the problem is and why it's important. They're going to summarize, again, that background literature, and they're going to describe the purpose, rationale, and design of the present study. And again, it's going to kind of lead to the hypothesis. So if I'm looking for a hypothesis in an article, it's not always the case, but where I start is I go to the introduction, I go to the last sentence of the introduction, and I kind of start to read backwards, because usually that's where that hypothesis is. They're going to spend the introduction introducing this topic and what's going on and what we know and what our question is, and then they're going to say, and here's what we think, hypothesis. And then usually they go almost straight into the methods from there. So it usually is in that last paragraph of the introduction. Not always, but it typically is, because again, they've spent the introduction trying to work up to telling us what their hypotheses are. So here's an example from Green and Flavell. They did a study, and this is again from their introduction, and they said the first hypothesis was that 5-year-olds would be able to report their thoughts if their thoughts were simple and familiar, and if substantial contextual support for having and reporting them was provided. So they tell us very, very explicitly what their hypothesis is. And a lot of times you'll see that, okay? Here's another. They had 2 hypotheses. Here's another. This is the background for where that hypothesis came from. So this would have been earlier on in the introduction. Estes and Harris suggest that young children may find it easier to introspect from visual images than other kinds of content. So because we learned about the work of Estes and Harris in 99, 98, and 95, Flavell and Green in 2000 made this hypothesis that given what Estes and Harris have taught us, that this is what we think is going to happen. So the stuff outlined in the blue is the background information that supports the hypothesis that's listed in the purple. Here's another hypothesis. This one had 2 hypotheses. So they have a second hypothesis, was that 5-year-olds would be less likely than 8-year-olds and far less likely than adults to notice the occurrence of thoughts that they did not expect to have. So they were going to be more likely to notice, the younger you are, the less likely you're going to notice a thought you didn't mean to have. So they said that again, that this research came from somewhere, this idea came from somewhere. And they cite their own work as well as other work saying that again, they do not realize that people are continually experiencing mental content of one kind or another spontaneously in this ever-flowing stream of consciousness. So younger kids aren't realizing this. They also say that that unlike the older individuals, preschoolers don't consistently attribute mental activity to a person who just sits quietly. So preschoolers don't realize that if you're sitting quietly that something's still going on. They don't quite understand that yet. And so both of these known results are what led them to make this hypothesis that the age was going to be a factor in determining whether they thought how their introspection task went. So when we read this, we need to really evaluate it. Did they do a good job reviewing the literature? Did they correctly represent works? Now, it's going to be a lot harder for you guys to do that piece of it because you don't have a big body of literature that you've read behind you. But if you have read articles that they cited, did they do a good job? Did they not? Do they talk about the purpose of the study? Do you see the critical thought that went into this hypothesis? Do you see the dots connected between the research that they've laid out and the hypothesis they've come to? Again, that's one of the reasons that this class is a critical thinking through writing class, is because this is what researchers are doing. They're showing you ... Here is the path and here's my hypothesis through their writing. And so this is a skill that's really important in psychology. And so it's a skill that it's really important for you all to have. Did they clearly state their hypotheses? And are those hypotheses testable, which is going to be a really big piece of the puzzle? On to the methods. So that methods has sometimes excruciating detail about what they did, right? Again, sometimes they're going to have it so explicit as to, I know in my own work, I talk about the brand of headphones that they listened to, the inches of the TV, how far away from the TV they were, kind of every minute detail, so that you can really kind of picture the space that this study was conducted in, and have the information, the specific information necessary to replicate it yourself. They're going to talk about independent variables and dependent variables, and they're going to talk about the operational definitions for both of those. They're going to talk about the measurements, and what's important for you to think about when you're looking at their measurements. Are their measurements measuring what they hope to measure? Remember, being a consumer of research, part of that is being skeptical. So really kind of look at this and say, okay, are they doing a good job of this? Are they going at it the way they think they do? And again, the purposes describe in sometimes excruciating detail how this was done, so that they can replicate this study. There's subsections under methods, and those really vary depending on the kind of study that they're doing. Typically, you'll have participants. Pretty much all of the research that I've seen come out of that 3530 students have found has been with humans, and so we have a participant section. So you'll have that information about who the participants are and all of that, the materials or the apparatus that was used, and the procedure of how it was used. So here's an example of a method section. They tell us these are two different ones. The infants, there were 72 infants between 12 and 18 months of age, but they also tell us the age, the mean, okay? So they give us the range and the mean. They give us two measures of central tendency, two descriptive statistics. They tell us how they got them. They were recruited from a large metropolitan area in a small city. They talk about the demographics, white and middle class, and they talk about that they had not seen these target DVDs ahead of time. It talks about any infants that were, in this case, children that were excluded as well. And they didn't just say children, they just didn't say infants, they were specific about what age. In developmental studies especially, that's going to be very important for you to include in your summary. But in every method section, they're going to tell you about the age of their participants. Again, we have, here's undergraduate samples. So 104 white undergraduates participated for partial credit. We still have the age there, and we have the breakdown of male to female. So we have 68 females. We have the mean age was 19 years old. So again, methods are part, they start with this participant because, again, we're dealing with human subjects. It tells us these are the kinds of things that are included, the breakdown of men and women or boys and girls, the average age, demographic information, and how they were recruited. The procedure is really the most critical component to some extent because, again, this describes what happens from beginning to end. This is where our replication information is. And again, that really sometimes excruciating detail. So here's a procedure section. So they talk about the participants, how they were recruited, what they were doing, that the Confederate entered the lab and how many minutes, the demographics of the Confederates. This was a study that was looking at kind of race in opinions about things. And again, it gives an overview of what the participants went through to participate in the study. Talks us about where it took place, who the experiments were, the order of the measurements and manipulations. And again, there's enough detail, not necessarily in this paragraph, but in the procedure section as a whole, that I could go back and replicate this study. So again, our consumer of research, let's evaluate those methods. Who were the participants? Was that going to be a representative sample? Are there issues with that sample? Kind of think through those things. Does this design allow an adequate test of that hypothesis? Are we testing what we sought to test? Do you see flaws? Do you see confounds? Do you see problems with the materials or the procedures? And sometimes this is a good place to allow you to kind of map out the study. Students also really tend to get stuck here when they're coming up with their criticisms. They like to talk about the sample not being representative enough and really say, next time they should do this study with more people or a more representative sample or more races. And yes, they should do that, no question. But that doesn't really require much critical thought on your behalf to really say those things, because we kind of always know that we're always going to want to have a more representative sample. So really don't get stuck here. Really use your thought processes and what you're learning in research methods and what you know from 3510 to really evaluate these things so that you can really use your critical thought and really come up with issues or changes that you would suggest. The results tell us how the data was analyzed and if the hypotheses were supported. So this is where they're going to tell us if there a difference between the groups. The treatment group performed better than the control group. That's the kind of results you're going to have. And they'll say they significantly performed better, and this was calculated using this kind of test, and they'll give you all the numbers. But this is telling us where the difference are. What's important for you to look at is, did they note those major findings? Did they find differences? Did they support or reject their hypotheses? Were they able to reject the null or not? Again, this is kind of that climax of that research support. This is why they have written and done everything up to this point to get this information. And this is really where the other part of 3530 is going to come in when we start to do the statistics as well, is going to be really evident in this results section. So again, they're going to answer those questions raised in the introduction, but they're going to be very stick to the fact. They're going to say there is a difference. They're not going to say what that difference means. They're not going to say this means that altruism, seeing altruism promotes altruism. They're not going to say that. That's going to be saved for the discussion. What they're going to really say is that the group of people that saw altruism performs differently than the group that didn't see altruism. And that's really what they're going to say in the results. Looking at the graphs and tables can be very, very helpful. In fact, if they have that, look there first and then look at the numbers in the paper. The other thing that's helpful is if they don't give you a table or a graph, you can make one yourself. So you can draw yourself one and kind of draw out the differences in means, because it is very, very helpful to kind of see these things and see the relationship between these variables. Or the levels of the independent variable. You can also draw greater than or less than signs between the different means on these graphs to really kind of help you figure out which one is doing what. So here's an example of a results section. They start out, no differences were obtained as a function of gender, so they didn't look at gender. So they looked at it, saw there was no differences, and so they stopped looking at gender, is what they're going to tell us. Then they go in and they talk about the other information. We're reminded about how the data was coded. We're reminded about what kind of statistical analyses were used. And those findings will always start describing what kind of statistical test was used. So here they tell us that they're using a 2x3 ANOVA, which you can see right there. So in a 2x3 analysis of variance, ANOVA, it indicated that the manipulation was significantly influenced participants' mention of race during the interaction. So they found a significant effect. And then they have their F statements. So we have an F statement saying here it was significant at 0.001. Less than 0.001, and it also gives us information about descriptive statistics as well. We have information about standard deviation and those sorts of things, in addition to those inferential statistics we see with that F statement. Again, those figures are really, really helpful. I highly recommend looking at those. Just by looking at this, you can see which one performed best and which one performed worst, and you can kind of look at that ahead of time. Another thing that's important to notice are these error bars on these graphs. Those are the little bars that go from the top of each of the bars, and they are telling you how much error is in that data, how much variability is in that data. And so you see here that the error bar for video with no interaction basically fits inside the error bar for control and almost fits inside the error bar from video with interaction. And so you're not going to see any, without even looking at the statistics, I can tell you that you're not going to see any differences really between those three. And if you do, it's going to be very, very little because they all overlap, right? Whereas parent teaching performed better, and that error bar is almost completely outside of the range of the other three. So we're really going to see that parent teaching is going to be very different, and we can see that by those error bars alone. So I don't even have to look at the numbers. I don't even have to look at the values to really see those things, and that error bars gives you that information. They also all have captions that tell us about what's going on here. So this one is telling us that children's mean performance on the post-test as a function of group. So this was the dependent variable was the performance on the post-test, and the independent variable was the 4 different groups. And it says that each child was tested with individual set of target words, and that the error bars represent the standard errors of the mean. So again, it's telling us about this information as well as labeling the various axes as well. So when we evaluate these results, we need to see what effects are statistically significant. That's one of the things it's going to tell us. Were the difference reported large or small? A lot of times you'll also see measures of effect size. Pay attention to that. Did they use the right kind of statistics? Were there errors between their text and their tables? Did they get it all right? And again, if there's not a table or figure, draw one yourself so you can really see what's going on. And remember, the results are telling us that there is a difference or there's not a difference. Discussion is, again, giving us what that difference means. And again, it's a clear and concise statement of those essential findings. So how it either refutes or supports that hypothesis. This is what that interpretation of what this means happens. So they're going to talk about these different things. And they're also going to talk about any limitations or explanations for weird results, these different kinds of things. So here's a discussion section, and this was of the 12 and 18-month-old infants. And this was a study that was looking at kind of basically watching baby Einstein videos and seeing if they learned words, which they don't, and that's a whole nother story for a whole nother time. But so they found that this is a clear answer that between 12 and 18 months, they're learning very little from these products that are promoted to this group. So they're not saying, that there was a difference in the groups. Here they're talking about the implications of, what those differences mean, okay? So they're re-summarizing those results in kind of the normal speak, just how you would tell somebody without kind of any of this jargon that's more specific to psychology. Sometimes if you have multiple studies, they'll also have a general discussion. You'll have a discussion for each experiment, just like you'd have a methods and a results for each experiment. And then to sum all of the experiments up, there'll be a general discussion at the end. So here it says across the 4 studies. So again, they use this general discussion to kind of summarize all the studies together and look at the implications and the limitations of that. Again, sometimes they really lay those limitations out in a limitation section. Not always, but sometimes they do that. And the kind of issues you could have would be issues with your sample, experimental design, manipulation problems, measurement problems. And it's always, the researchers are going to want to pull those out themselves and say, hey, this could have been different. I could have done this better. Because that really is saying, this is what I think is happening, but these other things could be an issue. So we need to kind of take it with a grain of salt. Again, they talk about the broader implications of these findings. These really are conclusions. So again, sometimes there's not a specific conclusion section. Sometimes we really just have a kind of the implications talked about in that general discussion area or in that discussion area. But they talk about the implications. So one practical implication of these findings for intergroup relations is straightforward. In situations where race is potentially relevant, whites who think that avoiding race altogether will shield them from being perceived as biased should think again. Clearly, that's a very relevant finding that has very relevant implications, okay? In fact, there's some various societies and groups of people that really should read this stuff. So again, they'll give us these implications of what this research really is. So saying like, okay, this is what we found, and we supported our hypothesis, but this is what this could be used for. The conclusion, again, sometimes they give it to you in a section. Sometimes that's just included in the discussion as well. But again, it kind of goes back to these kind of more lofty ideas. It's very, very broad. It talks about those implications of the study and these sorts of things. So you need to evaluate the discussion just like you've evaluated every other section. Do you continue to see that logical flow of ideas from introduction through methods, results, and now into discussion? Do they make speculations concerning these? Do they mesh with the previous research and the previous theory? Or are they really kind of counterposed? Do they talk about those directions for future research? And then do the same thing for the conclusions. Do they talk about those broader implications? Are the implications logical? Do we continue to see that logical flow of ideas throughout? So again, the kind of distinction between these three, and I made a separate slide for this because students really do tend to get confused. The results are, is there a difference? Okay, psychology students have higher IQs than chemistry students. I made this finding up, but I like to think that this could be the case. Discussion, what the difference means. So what does the difference that you found in the result mean? Psychology students may be smarter than chemistry students, okay? And then the conclusion is the broader implications of that finding. Psychology may have great future scientists given the wealth of intelligence in the field. So again, you can see that we start out very, very narrow. We're just talking about IQ scores. There's a difference in IQ scores. And then we get more broad with the discussion that we're talking about intelligence, and we get more broad with the conclusion. We talk about future science implications. All papers will have a reference section, and all of your papers should have a reference section. And these are very helpful ways to find articles. So if you're reading an introduction and you find something, they're talking about an article that you're really interested in the introduction, they have a reference. You can go to the reference section, find what this article is, and go find it yourself. So all that information is in the references. And they also see this proper kind of citation and the proper references. You can look and see kind of how yours should look, okay? So we see hanging indent. We do that to really save space. So we can see who we're talking about. You can see where the new one starts, but we don't have to have spaces between these references then because of that hanging indent. So you don't need to, do an extra space after your first reference to start your second one because the hanging indent does this for this. They're always in alphabetical order. One of the reasons when I graduated with my PhD, I was already married and I had to decide, okay, do I want to be Kuhlman-Atchison or do I want to be Kuhlman kind of as a middle name and Atchison, my married name, as my last name? And I chose Atchison as my professional name instead of Kuhlman-Atchison, primarily for alphabetical reasons. if I'm the lead author on a paper, I'm always going to be towards the top with Atchison, whereas Kuhlman, I'd be kind of down in the middle with my K. So again, we're going to alphabetize the references. What you do not ever alphabetize is the order of the authors within the reference, okay? So for example, the Deloche in 2009, you can see Deloche is still coming before the Chiang, even though that starts with the C. And the reason that is because Deloche did more work than Chiang, okay? So you're not going to switch those because she did more work, okay? So it doesn't matter that you don't alphabetize the authors within a reference. You just alphabetize the references. You see the same thing down here with the Cremar, okay? Those are not in alphabetical order for the same reason. And even the ones that do appear to be in alphabetical order aren't alphabetized. They really just are. Anderson did more work than Pembick. That Hay did more work than Hebert or Simcock. So those are, again, it's telling you who did the most work on this paper, who's the lead author, who did the most work. Again, we talked about hanging indent. We're only using authors' last names and their first and middle initials. So you don't see any first names here. You don't see any middle names here. We just see initials. You italicize the journal name and the volume number. So you can see this here. We have American Behavioral Scientist, 48 is italicized. We don't have anything else italicized, okay? And that will include the issue number, which is confusing. You're only going to italicize the journal name and the volume number. And this is, again, the proper formatting from this. And this can be all the information on how to properly do this is available in those APA guides that I suggested getting, but it's also available on that Purdue OWL website. If you need help reading a research article, I know they're complicated. I know they're dense. I know this is hard. The Psychology Method Center is really available for you. It's a 55-minute appointment in Cal. You register for it on Sona. It gives you help with reading those primary articles. You really have to read this article prior to the appointment. You can't come and say, hey, let's read this article. No, you need to have read the article prior to coming in, and then the tutor will help you. The tutor will skim it and help you answer your questions about it, okay? And you can also see me for more help. I'll be happy to do this with you as well. So really, if you have questions about the content in your research article, please see me or your GLA. And again, the Psychology Method Center is a great help too. See you in class.